I think some people.
So there's a laminate which contains all the SNPs. So a coenzyme, if you remember, there's 20 coenzymes and there's 20 SNPs, permutations of SNPs. So in other words, there's a mismatch in the DNA bases in the wrong position. Okay, so there's 20 possibilities because you've got 5 nucleotide bases, and therefore you've got 4 can go into 5, which makes 20 possibilities. Okay, a bit like the National Lottery. Okay. So they encode for a greater need for a coenzyme. Okay, so to bring that whole biological pathway up to fruition. So in my case, mine was adenosylcobalamin, which means I need a little bit more adenosylcobalamin than what I'm actually making in order for my biological systems that use adenosylcobalamin to work to their best. This is how we overcome, because we can't change our genes, but we can epigenetically bring out the best. So this is what a coenzyme does. It does not mean I am deficient in B12. Okay? Otherwise, I would show to a vitamin. In this case, it would be hydroxycobalamin. Okay? I only show to adenosylcobalamin. So this means that I am short of being able to activate B12 to get it to work. And there's only several pathways in the body where this occurs, where we actually use adenosylcobalamin. So in this case, what we want to do, once you've got this, if you have a look on the side which is coenzymes. Now, the coenzymes are activated vitamin Bs. So we've gone through here, so we're looking at this. So the coenzymes are B1, B2, B3, B5, B6, folates, B12, biotin. And then the ones that are not vitamin Bs are alpha lipoic or lipoic acid, vitamin C, SAM, and CoQ10. Okay? So those are not vitamin Bs. All the other ones are vitamin Bs of different types. So a coenzyme is an activated vitamin B, or a substance that the body needs. You can't get activated vitamins from your diet. You get vitamins, and you have to make the activation in the intestine or the liver within. So it is a genetic problem that you are unable to make enough or get the substance to work. So on the other side, you'll see the genetic code. So at the bottom, there's that little table, okay? And the genetic code is very easy. Now, some of these polymorphisms have been known before other ones. So, for instance, certain ones involved with the folates were the first ones to be discovered with breast cancer, where you had gone into the T position, which encoded for methylene tetrahydrofolate. So there's a number of ones which have been known for a long time, and then we worked out the rest of them, because the amazing thing is, in genetics, they don't seem to be interested in doing anything about it apart from using stem cells and looking at proteins always, and not how to get the epigenetic side to get the person to work better. And the reason solely for that is there's no money in it. It's just purely money. There's no drug. Okay? If you say, well, you could actually improve a person's performance by giving them adenosylcobalamin, they won't even publish it. You know, this is just rubbish. You know, they have to have stem cell therapy or some form of genetic work. You know, they just think you're crazy. Nutrition is nothing to do with health whatsoever here. So what we're going to do is we're going to, we can leave the magnesium on at this stage because she still needs the magnesium anyway. So now we get our 5 DNA bases. So this is your adenosine, adenine, cytosine, guanosine, thymine, and uracil. This is our 5 DNA bases. And what Jill's going to do, she can leave the magnesium on, but no, obviously, colour on there. From strength, we're going to see which one weakens. There will be one which will weaken her.
Well, from strength, okay.
From strength
Take
to
the
see.
380 off, yes.
Take the 380 off, yeah. So say what you're doing.
Yeah, taking the 380 off, so we're in strength now. Check you in strength. Hold. Okay, now we'll try adenine. Hold. Strong to that one. Now cytosine. Hold. Strong to that one. Guanine. Hold. Strong to that one. Thymine. Hold. Strong to that one. And uracil. Hold. Weak to uracil.
So she's weakening to uracil. That means uracil's the bad guy, right? Uracil is a base which should normally be in DNA, in RNA. Sorry, in RNA. That means she's got a touch of RNA into a DNA position. So she's got something which she's not converting that uracil into an RNA. So now we leave that on, so she's in weakness, and we run through the other four now to find out which one negates it, and then you've got the code.
Okay, so adenine.
So we're now doing U into A, if you like. So she strengthens that one?
Yeah.
Yes. So you now look up there, U into A. Okay, carry on. You only find there's one.
Yeah.
So U into A is a folinic acid, yeah. So usually anything involving U is usually a folate of some form.
Hold.
Could you get some, have you got some folinic acid over there? Folinic.
So just the A. So U into A.
Yeah. So the answer to her polymorphism and her genetic aspect of the 380 is folinic acid, is U into A. Do you see that from the code? Yes, okay. So on the code at the bottom, U weakened, A negated. Therefore, it means that U's gone into A, and that's the SNP. So you just look up the code then, what it is, and it encodes for, in this case, folinic acid, yeah. Yes, we left it on because that was her sort of preliminary thing. Yeah, yeah. So initially, she went straight into triple warmer, and then the reason I've gone down this is because she showed up to be needing a coenzyme. So always when a person shows to a coenzyme, it's an indication they got a SNP. If she needed folic acid, she would show to a vitamin. If she was deficient in folic acid, folic is a vitamin, folinic, 5-methyl tetrahydrofolate, etc., are all coenzymes. So now we just do the dose in the usual way. So you can take off the nucleotides and just use the 380. As we said yesterday, there are some strange things with coenzymes that sometimes, and we're having to now extend our product kits all the time because sometimes people will show to a dry powder capsule rather than a liquid, and some people will show to a liquid rather than a powder. So the genetic code here, in my opinion, is always right. Okay, so if you take that as gospel, okay, and if they don't strengthen to the product, it's because the product is not right. Okay, so the product is either not pure or it's not the correct form. It might be a capsule rather than a liquid. You know, it confused us for ages, you know, why this is. We came to the conclusion, as I say, that with B12, it's a matter of the intrinsic factor, as I described yesterday. That if a person swallows a capsule, the capsule will stick to the stomach will then produce hydrochloric acid, and with that it produces intrinsic factor because they come from the same cell. So you're more likely to absorb a capsule in this circumstance, perhaps for somebody who has a difficulty in absorbing B12. But as a liquid, in some people strengthen to the liquid. But remember, generally a liquid will go through the stomach very quickly, particularly if it's diluted in water, and may not stimulate enough intrinsic factor to be able to be absorbed for later down. Okay? So sometimes a capsule will work well with a person and sometimes not. Pyridoxal 5-phosphate appears to be the same. So I think pyridoxal 5-phosphate, when you take it, does need some intrinsic factor involved with it, and I think probably B1 does as well. So we're having to do most of our coenzymes in liquid form and in a capsule form, okay? Just so we batch it both ways. So assume the genetic code is not wrong, okay? It's you who's wrong or the product is wrong. Okay, how are we doing?
Yeah, three capsules.
3 capsules of folate.
Yeah.
Now, when do you take any folate? When do you take it? last thing at night. Okay, why? And because folate, although it's an acid, is a very weak acid, it doesn't like hydrochloric acid. Okay? So if you pump it into food, you take it when you've got food and you've got high hydrochloric acid, or in fact you've got higher hydrochloric acid between meals, when the stomach is actually empty during the day, is the time when hydrochloric acid is highest. You don't want to swallow the folate because it's insoluble in acid. When we make a liquid folate, we have to put a little bit of carbonate in, either magnesium or potassium carbonate, just to alkalize it slightly to get it to dissolve. So that's how you would absorb it. So you always take folates last thing at night. Okay, so there's no acid in the stomach there. Right? That applies really to 5-methyl tetrahydrofolate, folinic acid, methylene tetrahydrofolate factors, or straight folic acid, tetrahydrobiopterin, anything folate is it. So there's a couple of other things we might as well just say at this time, if you remember, that also you take last thing at night. And you can simply check this by just going around, the different meridians, as we did with the body clock, and finding when to do. So CoQ10 is another one. CoQ10 hates acid. You just swallow CoQ10 during the day, again, complete waste of time. Which it is, it's a complete waste of time taking CoQ10, in my opinion, anyway, unless you show you need it. Just taking it for the sake of taking it, I don't think is a good idea. Because all these coenzymes you should make yourself, as long as you've got the raw vitamin there. If you take a coenzyme and you don't need it, you're going to shut the pathway down of actually being able to make it yourself. So I don't think you should take CoQ10 or any of these if you don't show that you actually need it. Treat them not as medicines, but treat them with greater respect than just a vitamin or a mineral deficiency. Okay? So where have we got to now? We've got
3 folinic acid.
So the other thing last thing at night, very, very important here, is a probiotic. Again, remember, between meals, your stomach acid is higher than when you actually got food in your stomach. Your food is actually absorbing some of the acid, so the pH is actually higher when you've got food in your stomach than between meals, and particularly just before a meal, because your stomach is then burning to have some food in it because it's secreting acid. Do never take a probiotic then, because you'll just destroy it, which is what the acid is designed to do, is to destroy bacteria. There's other functions with it, but one of them is it's antimicrobial. You must always take a probiotic last thing at night, and particularly the one we're going to talk about later on, which is one of the most important probiotics, which should be in the human body, which really isn't in the human body enough to make such valuable commodities as vitamin K2. So we're missing Bifidobacteria subtilis, which is the principal one in ruminants. So this is the one that ruminants have in their cecums, and that's how they make K2 and probably B12 and another load of other nutrients, biotin, pantothenic acid, these ones which are less essential nutrients because you get them from bacterial fermentation, if you've got the right bacteria in your gut. So most of us do not have the right bacteria in our gut. We're pretty sterile of the ones that we really need for 3 reasons. One is antibiotics. When someone takes antibiotics, they destroy all the good guys in there. The second is chlorinated water. Okay, and the third is food preservers. And probably the food preserver is a bigger problem than all the others put together. Because people have preserved food almost every day, haven't they? Something which is preserved. And if it's got a preserver in it, it preserves the food, but it preserves your gut as well, okay, from the good guys. And the more guys you got in your gut, the better you are, the healthier you are, and the more variety of good guys you got in there, the better you are as well. So they found, many, many problems with people who haven't got the right type of flora or have too little amounts of flora. They need more dirt in there, like children grovel around, and the more dirt that goes in here, the more probiotic or good bacteria you've got, the better their health. And one of the things we've discovered, and we'll be covering that probably in the next module, is about the carboxylated amino acids, the things like tyramine and things, which can be so toxic to the human body when these proteins aren't digested and they're decarboxylated and they form all these poisonous byproducts in the gut, like cadaverine and putrescine and all these, and they create so many mental issues. They affect the brain chemistry like mad, and And they're all there because we don't have the right probiotics in our gut. So getting the right probiotics, and the most important one is that B subtilis. But we're going to talk about that later. Okay, so do you want to just do a quick challenge without anything on her of her scales? So let's see what her scale of health is. I think her scale of health, my feeling there is it's going to be quite good, with 100 being the best she has been. Would you agree? Even though she's run 40 miles. Okay, let's try her anyway. So the scale of health.
Epigenetic scale of health?
Yeah, no, the scale of health with 100 being the best she has been, the regular scale of health.
Okay, so on a scale of 1 to 100, your scale of health right now calibrates at 70 or more, 80 or more,
Ooh.
70 or more, 75 or more.
Not as good as I thought.
70.
Okay, so
70.
she's about to run to America or somewhere, run across America. At 75, 70 is it? 70,
yeah.
Yes, okay, now do the epigenetic. So let's see what her foundation is.
Okay, so on the scale of 1 to 100, your epigenetic scale of health.
With 100 being the best she could be.
The best you could be. Your epigenetic scale of health calibrates at 50 or higher, 60 or higher, 70 or higher, 80 or higher. So that's
Yeah, 75.
75 or higher.
Yeah, so she's coming in more or less the same, isn't she? About 70 on both. Now shove all the good things on, the folinic acid and the magnesium. So that's good.
Oh, these test well.
Finn, what you have to remember when you put those goo dishes on, right, is if there's two lots of white capsules and they're the same size, and you take them off and you think, oh no, which one is which? So you have to have some other. If they're different colours or anything, it's okay, isn't it? But I've done that so many times. I've taken them off and thought, oh no, which is which? You know, and then in the bin.
So on a scale of 1 to 100, your epigenetic scale of health right now, to be as good as you could be, calibrates at 70 or higher, 80 or higher, 90 or higher, 100, 110.
Good. Oh, yes, that's right. Actually, an athlete does want to be 110, really, don't they? They don't want to be 100, they want to be 110. And her normal scale of health, her regular scale of health now, with 100 being the best she has been.
Okay, on a scale of 1 to 100, with 100 being the best you have been, your scale of health right now calibrates at 70 or higher, 80 or higher, 90 or higher, at 100, at 110.
So that's pretty good. We've got to back up, thank heaven, because if she's going to go running next week, you know, and she needs, 4 magnesium and 3 folates.
Yeah.
Okay, remember the folinic, in this case, is the base. It's the inherited foundation. So to make folinic, you probably need magnesium. So, you know, the need for the magnesium is down there. Plus, she's just probably lost a lot because of all the running. Okay. If you could go back onto the screen. So functional genomics. Yesterday we talked about the chromosomes. If you remember, this is, what page is this? Oh, you've got it there. 113. If you remember, we talked about haploids and diploids. Haploid was the germ cells, and diploids was when you have a pair of chromosomes here. So inside the chromosomes, we can see we have ultimately our DNA ladder, which is uncoiled with our base pairs of the 4 nucleotide bases, and in the case of RNA, uracil as well. So those are the bases that we looked at. There's not much point in testing with the nucleosides or the nucleotides, which is what actually occurs in the RNA structure, DNA structure, because you can use the bases. It's the bases which we want to know what the mismatch is. And the mismatch is very easy. The one that weakens is the bad guy that's got in the position of the good guy, which is the other one. So each of those has a code to it. Here's a strand of DNA. You can see that it's the nucleotides here. This would be cytosine here because it's got a C on it. You see it's then got a ribose, sugar added, and a phosphate group. And with that phosphate, you see an oxygen there, held suspended. It's that oxygen that gets oxidized and becomes a free radical when you're exposed to reactive oxygen species. So that's done when the free radical does the damage to DNA. It's really, really very important, because if it damages DNA and oxidizes that, the DNA then can't be read. So what has to occur is an enzyme has to come in here and cleave or cut this out and replace it with another one, okay, a new one. So every time we oxidize DNA in any way, which occurs, if you remember from what was said, there was 25,000 to 100,000 hits on every cell per day. I guess there's a lot of repair going on. These have to be cleaved off or cut out and a new one put in, inserted into it. Bruce Ames, who said that. Now, if you don't have the enzyme to do this, then you don't necessarily put the right one in, and the ladder collapses, because these are like the sides of the ladder. So you have to put in the exact one. Otherwise, if you put something else in, you get a mutation, or single nucleotide polymorphism, as it's called. Now, the enzyme that does that is called DNA polymerase. DNA polymerase. And DNA polymerase is a zinc-dependent enzyme. Now, zinc is used a lot in the body, but its main function is DNA polymerase, which means it's the repair of tissue. So if you're deficient in zinc, the place you see it is that you don't repair. Okay, this is the most important thing for us as practitioners of dealing with people's health conditions, is our patient repairing? They've had an injury, or they've got a reactive oxygen species inflammation. Are they not repairing? So the most important and most common mineral that people show to be deficient in is zinc. Now, I think you'll probably all agree there, of all the minerals that you probably prescribe, zinc would be number one. Well, that's in my case is. And magnesium would be number two, and selenium phosphate certainly would be number three. After that, the rest of them are sort of way down the list. But those three are the most important minerals that there are, and mineral compounds. The next question is, which type of zinc? Because again, very interesting, isn't it? And that some people will strengthen to only one, or most people in fact, will only strengthen to one form of zinc. You know, sometimes people will strengthen for two or three, but they don't strengthen to all of them. You know, we do a chloride, a sulfate, a picolinate, a citrate, and a ascorbate. And if a person was zinc deficient, you'd think they would strengthen to every form of zinc. But I think they use different parts, different types of zinc in different areas in the body. So you may find that one zinc works well in one area of the body, and another works in a different area in the body. Or it's to do with the absorption for that particular person. So we also do a triple zinc. We do a smart zinc which contains celery, which helps to get it activated. But we also do the same one without the celery, which we call triple zinc. So that is basically ascorbate, picolinate, and citrate. And that's very popular as well. You know, a lot of people say, well, that's the only one that really works with them. So again, you have to test different types of zinc. It's no good just saying, oh, you zinc, you need zinc, and just go run down to the health food store and buy yourself some zinc, which is usually zinc gluconate, which doesn't really work for anybody. You have to tailor it to the individual requirements. And remember that when they do strengthen, then cross-check it to both meridians, both sides of the body, to make sure it is the right one entirely there. So DNA polymerase, so whenever zinc comes up with a person, think, they have a problem with DNA polymerase. And that is where you repair the DNA in the nucleus, but also we have DNA, as I've taught in previous modules, in the mitochondria. The mitochondria has its own DNA, and it has hundreds of DNA molecules in a mitochondria. And those mitochondria DNA are circular. So whereas nuclear DNA is like a ladder, is linear, in the mitochondria they're circular, and they're just like a bacteria. And that's why they think somewhere many, many billions of years ago, anaerobic bacteria were invaded by aerobic bacteria. And that's what we've been left with. The mitochondria is actually an aerobic bacteria when the Earth's atmosphere was changing from methane into more oxygen, and the oxygen or aerobic bacteria were on the ascendancy. So this is a theory, and it may be right, may not be right. So the mitochondria has its own DNA, and it's circular, and it encodes for all the enzymes in the mitochondria. So it's a self, repairing mechanism. So as long as you have the DNA, the zinc for the DNA polymerase, the mitochondria can look after itself and repair itself. So it encodes in there for all the enzymes of the Krebs cycle and for the complexes 1, 2, 3, and 4, and 5 in the mitochondria itself and all the cytochrome enzymes that are involved in them. So it takes a lot to damage that lot. But there is a lot of oxygen inside the mitochondria, and there's a lot of energy being produced and a lot of electrons, because we're getting 4 pumping into that membrane with every equation. That means we've got a lot of free radicals being produced. So this is why we have a lot of DNA in there to repair. And DNA damage in the mitochondria is now being looked at much more importantly for a lot of neurological problems. So a lot of more serious neurological diseases and things, the research is going into mitochondrial DNA with neurological problems. And one of the most important ones there is Alzheimer's or memory problems, which we talked through at various seminars through the year. So we now know that probably the death of the neuron in Alzheimer's disease is largely caused from mitochondrial dysfunction. And the neuron basically is not generating enough energy, and so the energy is needed to pump out the calcium and sodium, and so the cell dies and the brain shrinks. So understanding about DNA and mitochondrial DNA is really very important, and how to build up the energy. So when a person is low in energy and they're hypoxic, they've got the dreaded 2 things that we saw. If you've got low ATP and you're hypoxic, you're just going to burn your mitochondria out. So you've got to really help to rebuild that up. So whenever whenever zinc shows up, think, we've got a repair problem. So here's the normal basing, A to T or T to A, C to G, and G to C. So these are the normal rungs of the ladder. If we do damage to the ladder, to the sides of the ladder, through oxidation, the ladder, what they call a nick, the ladder will not be structurally sound. If we get a nick on both sides, so in other words, you've got an oxidative damage on one side and on the other, the ladder collapses. So your DNA is finished. So in other words, you can't repair. You can only repair if you've got a nick on one side, because you always model it on the other nucleotide on the other side, like a mirror. So T will always pair with A will always pair with T, or should do. So if you've got damage on one side, you mirror it from the other one and you replace it with the DNA polymerase. But if you lose both, you oxidize both these, and then the genetic code can't be read. And the ones that do that are the peroxynitrites and the hydroxyl radicals, because they're much more oxidizing than the other ones, the superoxide. So we know with the genetic code that we have a coding strand. One of these strands is a coding strand, and the other one is the template. So what we always do is we always model the coding strand here to the DNA. So on the coding strand of the DNA here, we've got this part which is A, T, and G. And you'll see on the RNA, which is the coding from that, will be A, U, and G, because U replaces the T, or the thymine is always replaced by uracil. So you notice A, T, G there on the coding strand of the DNA, which transcribes into A, U, G on the RNA. Now this is interesting because this code, ATG, is the code for the amino acid methionine. Okay? Now every genetic code starts with methionine. So a genetic code will always be just 3 codes, 3 nucleotides there, and then another, and they always start with methionine. So this could be hundreds of amino acids long to make a protein, but they all start with ATG, okay, or methionine. So methionine is the capital letter at the beginning of a sentence. There's no gaps between the words. So every word has 3 letters in it, and there's no gap. And then the sentence finishes with a full stop. So here we have ones which code for full stop. So when the genetic code finishes with ATT, TAG, TGA, etc., that's a full stop. So that's it. That is the genetic code for that particular protein. So we've got to obviously have the proteins. The genetic code doesn't make the proteins. It encodes for the position where the proteins go. So when it comes to amino acids to build it, you've got to have enough amino acids in the diet to be able to do this. And this is a question, is do people have enough of the right types of amino acids in their diet, in their pool, to be able to make the repair of the tissue? So this one, which is an RNA coding, UGC, which within the DNA would be TGC, encodes for selenium cysteine. So selenium cysteine is an amino acid where the sulfur on the cysteine has been replaced by selenium. And this is how the body uses selenium as an amino acid called selenium cysteine in the amino acid sequence of a number of enzymes. The thing is that the body cannot take selenium cysteine and use it for the proteins. It has to make it. And in order to make it, it has to make it from one substance, and that's selenium phosphate. It can't make it as selenium, or can't use it as selenium cysteine, so it has to make it from selenium phosphate. So the phosphate comes from ATP, adenosyl triphosphate. So if you're low in energy, you can't then activate your selenium to make the selenium-dependent enzymes work. So selenium can be in the form of a sodium selenate, which is the best ionizable form, but it has to be mixed with ATP and water to make selenium phosphate. And then the body uses the selenium phosphate to make the selenium cysteine by the RNA. So it's very clever. But taking selenium cysteine doesn't seem to be, the body apparently can't use it. There's muscle test to it, but it can't actually use it in making selenium-dependent enzymes.